University in Halle, Germany, and graduated there. And he worked in the lab of Thomas Lahaye, so which is also kind of famous in the TAL transcription activator-like effector field. And then during his PhD, he also worked with Ulla Bonas at the Martin Luther University. And currently he worked with Alain Tissier, so which is at the Leibniz Institute of Plant Biochemistry, which is also still in Halle. So, and he expanded the repertoire of programmable tools for nucleic acid targeted systems and their application for synthetic biology. So the title is already there. So Tom, many thanks that you share today your results, and the stage is yours.
So thank you very much. I hope you all can hear me well and see my screen. Yeah. Can you hear me?
Yes, we hear you. Okay.
Oh, good. Okay.
Yes.
So thank you very much for the invitation and the opportunity to present our data here, which is about engineered CRISPR endonucleases that favor homology-directed repair in plants. So I think I don't have to convince the audience that with gene editing tools like CRISPR, we are now able to generate desired traits within one generation. So for example, replace an allele or knock in a resistant gene. So you can, in the end, do the same thing with conventional breeding, but it takes a lot of time, including crossing and multiple back-crossing steps, that you end up with a trait that only carries a tiny snippet, and in this case, a resistant gene from a wild species into an edit line. So this is also our goal, to develop tools which can do so in a highly efficient manner. So as I said, we can do programmable DNA double-strand breaks. So we can cleave one locus in the background of a whole plant genome. And this we can do with, for example, CRISPR endonucleases, and most prominent is Cas9. So just briefly, it's a two-component system. So we have Cas9, the protein, the endonuclease performing the double-strand break. And we have a second component, RNA moiety, a guide RNA, so they can form a binary complex, and then they can bind and cleave DNA specifically. So the specificity is defined by the first 20 nucleotides of the guide RNA, the so-called spacer, and it's complementary to the target sequence. And one prerequisite is that the DNA has a so-called protospacer adjacent motif, and for Cas9 it's NHEJ. So this is a specific contact of the protein to the DNA, the PAM, and the rest is defined by the spacer of the guide RNA. And the beauty of the system is, if I just want to target another locus, I just change the very first 20 nucleotides of the guide RNA and can bind another target. Again, the prerequisite is to have a protospacer adjacent motif close by. So how is this working? On the right panel, you see a structure view of the cleavage active Cas9. And what you see here on the left panel is my more easy model, let's say. And you probably know Cas9 has two endonuclease domains. One is HNH, which is responsible for cleaving the targeted strand, and RAFC for cleaving the non-targeted strand. So cleavage occurs orchestrate, and they both cleave 3 nucleotides upstream of the PAM, so they mostly produce blunt ends. And blunt ends are precisely repaired by a non-homologous end joining in plants. So what it does, it just put these things together, and there's no need for processing, so we can restore the cleavage site and then just go in the next round of cutting by Cas9. So RAFC also has a certain flexibility in terms of cleavage. So the non-targeted strand can be cleaved by RAFC PAM distally shifted up to 3 positions, but mostly 1. And if the shift happens, then you do not trigger a blunt end, you trigger a short 5' sticky end. And in this case, non-homologous end joining either chop off or fill in this overhang to again come to a blunt end, which then can be ligated again. And here you end up with a +1 base pair or -1 base pair insertion or deletion. And with this, the target sequence is destroyed and you have a stable edit. So these lesions, which are produced by Cas9, these are mostly substrates for non-homologous end joining here, a process that, as I said, just put things together. It allows minor processing, and if there is processing, you will end up with some mutations here. You cannot really control the outcome. That's why this pathway is considered to be error prone, and it's perfectly fine for making knockouts. But if you want to have a desired edit, it's not suitable to use this pathway. There's also a second pathway called homology-directed repair. So it's based on homology, and this always goes along with 5' resection. So which means it triggers, so these are lesions which have longer single-stranded 3' overhangs. And these 3' overhangs can serve as primer and anneal to complementary sequences, either to single-stranded DNA or invade double-stranded DNA. And there they can be extended, or the 3' end can be extended by DNA polymerases. And with that, you can copy a sequence from the donor into a specific locus of the host genomic DNA. When you look at the development of the recent years, so the last decade, let's say, so there are fancy tools which are all really good working, so base editing and prime editing, and also eventually CRISPR-associated transposons. But these are editing tools which do not trigger a DNA double-strand break, because the people think we do not want to lose control, so we rather don't introduce a double-strand break. Although they are really nice, they come along with some limitation in terms of editing. So on the one hand, you have a restriction in the size you can change, and also you need usually a target close by to the edit in order to realize these edits. So we followed a different approach, and it feels a bit like a step back, because we still want to produce DNA double-strand breaks, but we do not want to create lesions which are substrates for non-homologous end joining. We want to have lesions that have longer 3' single-stranded DNA overhangs, so substrates for homology-directed repair. And in order to do so, we just equipped CRISPR endonucleases Cas9, which produces blunt ends, or Cas12a, which produces 5' sticky ends, with 5' exonucleases. So we cut and 5' resect. In our lab, we have a colleague, Sylvestre Mayonnet, you probably also know, and he made a nice Nicotiana-Bentamiana reporter line, which is very helpful to analyze our constructs. And I want to quickly take the time to introduce this line here. So tobacco mosaic virus, so short TMV. So it's a transgene with a genome of the TMV inside, and usually you have 3 genes on this genome. So you have the replicase, a movement protein, and the code protein. So this transgene has 2 modifications. So first, the replicase has a 3.8 kb deletion, and instead of this 76 base pair outside. So the replicase is not there, it's destroyed. There is no replication going on. The second change is that the code protein is exchanged for GFP, and this has a benefit that this virus is not infectious anymore, so it cannot systemically spread, and we can monitor ongoing replication by just following GFP fluorescence. So what we did, we used this line to transiently express Cas9 and guide RNAs. And here I want to say that we applied guide RNAs in a dual guide RNA approach. So we have 2 guides, and we applied them either in the PAM out orientations using this combination, or in the PAM in orientation. The rationale was to find out some influence on the PAM, but you will see later that there is no. And in addition, we also delivered a tDNA carrying a donor, and on this donor, there's a complete replicase sequence. So after cleavage and repair by gene targeting, we can restore the replicase and initiate replication of the virus. And to cut a long story short, when replication kicks in, there's also production of GFP, which you can monitor easily. And the benefit here is also that the movement protein is present. So imagine that a single cell event in which editing happened can spread to the neighboring cells, and we can just macroscopically monitor raising GFP spots. And this is a value for the efficiency, for the gene targeting efficiency, as depicted here 6 days post-inoculation. So here's an overview, and let's start with the control. So as a control, deactivated Cas9, so there is no DNA double-strand break. There will also be no repair, so we don't see any signals here. And with Cas9, we see after 3 days, it's barely maybe a single spot, but after 6 days, we can see a few spots here. And if you combine this, compare this now to our exonuclease fused Cas9 variants, you see that among the candidates, we have a few candidates, which leads to a massive increase of gene targeting. And you see it quite nicely after 3 days here. You can barely see something for Cas9, but for UA12, this is an exonuclease from herpes simplex virus. You see a lot of spots are already there. And after 6 days, when you start to see a few points for Cas9, the system is already saturated here. Another candidate is a T7 exonuclease, which also gives a bit increased activity. And you can also quantify this here by just counting these GFP spots of defined areas. And we see that we end up with around 20 to 30-fold increase in gene targeting efficiency comparison UA12 use Cas9 and Cas9. So not all exonucleases work equally well. And we were also wondering why this is the case. And the easiest explanation might be that there's some effect of on expression rates. So we did Western here, and we can see that except for EXO1 fused to Cas9, that all full-length proteins are expressed and we can detect them. And there might be a reduced expression rate for UA12 fused Cas9, but it's still enough to trigger this high gene targeting efficiency. We also wanted to know whether there's precise repair. So we just genotyped by PCR. So we isolated genomic DNA and made PCR reactions. So we amplified the 5' junction here. As you see here, you don't see anything for the controls. But if you have cleavage, we see this. And the bands is thicker for UA12 and T7, which is correlated to the in vivo activity we see. We did the same for the 3' junction, and here we are already at the detection limit, but still for T7 and UA12 and even T5, you see the junction. These are all sequences, it's all precise, it's all fine. And another thing, we wanted to know what is happening on the target. So here we use primers that directly flank the outside, so the elongation time is too short to amplify an integrated donor, but we can amplify this sequence here. You see quite small fragment. And what you can see in terms of Cas9, there's a second band appearing, which is the result of the deletion of the fragment between these two cleavage sites here. And this band then in the end is non-homologous end joining because it just takes these two ends together. And we see that the second band disappears, and the disappearance is somehow correlated also with the activity. So UA12, it's almost gone, and also T7 exonuclease. So we wanted to further have a look into this and focused only on these 3 exonucleases here. So 2 of them are working. T5 exonuclease is not working as such as the others. So we repeated this experiment, and this time also without the donor. And you can see that this second band disappearance is independent of the donor, or the presence of the donor. And we wanted to find out what of mutations we have here. So we did amplicon sequencing, and what you see here is just a portion of mutated reads of all reads. And here's only focusing on deletions. And you can see nicely that Cas9 usually only makes deletions between these two cleavage sites here indicated by this rectangle. And if you compare this to the exonuclease fused variants, you see that we have some shoulders here, so they can resect a bit of the end. And if you see the maximum length, we see it's more or less comparable between these 3 exonucleases, around 60 nucleotides. But we can see that for T7 exonuclease and UA12 exonuclease, we have a overall higher number of deletions, and this is maybe correlated then with a higher activity, that we can resect these ends more efficiently than T5 exonuclease. Okay, we wanted to have a closer look for this because, as you know, and as I said in the beginning, Cas9 is producing usually blunt ends, and we wanted to know how these exonucleases can deal with blunt ends. We put in vitro assay, which was published before here. And to cut a long story short, if these exonucleases are resecting the substrate, you have an increase of fluorescence, which you can measure over time. And we can see that T7 exonuclease and UA12 are really fast in digestion or resecting this substrate here. You have a fast increase of fluorescence, whereas T5 exonuclease takes a longer time to degrade this substrate. And this is also correlated with the activity we measure, and it's also correlated with a literature because it is known that T5 exonuclease somehow disfavors blunt end DNA substrates. And this might be the difference why T7 exonuclease and UA12 is better than T5 or the others. But we can also see that in the vitro assay, T7 exonuclease and UA12 have more or less the same activity. But here in the in vivo assay, I would say UA12 type is always a bit better. And we wanted to find out, okay, what makes the difference between T7 exonuclease and UA12? So here I want to give you an extra information. So UL12 is kind of a special exonuclease. It's a relatively large exonuclease, so it has a large exonuclease core, but in addition to that, it has also an N-terminal region, the first 126 amino acids. And it was also shown that these interact with DNA repair factors, more precisely the mRN complex, and we want to know, okay, how much the contribution of this domain is to the gene targeting efficiency. So we repeated this experiment, but this time we included 3 mutants here. And the first mutant is the catalytic dead exonuclease variant. So there's no exonuclease activity by exchanging one amino acid. And we see that the activity drops down more or less to the Cas9 activity. You can see also the quantitative results here. Sorry for this diagram. I left out everything which is not important. So here's Cas9, and this is here the exonuclease dead variant. We also deleted the first 126 amino acid to see the impact of this. And you see there's still high activity, but it drops down by half, let's say. So there's a contribution. But if we only use the N-terminal domain and fuse it to Cas9, there's almost no change. So we can conclude that the exonuclease activity is absolutely essential for this increase in gene targeting efficiency, and that the N-terminal region has a minor contribution to that. Okay. We then wanted to know, this is more for the application, so how we should design our constructs, because you probably know when you do gene targeting, you have a donor which is flanked usually by 1 kb homology arms, 5' and 3', which renders the donors quite big, of course. So we wanted to know what happens if we reduce this homology arms and we reduce it to 500 or 250 or 100 base pairs on both sides. And here we see that 250 base pairs seems to be fine. It's even better than the other ones. So we conclude that, or recommend that having homology arms of 250 base pairs is absolutely fine. And this also is 1.5 kb less in size of donor, so which is also good for reducing the size of constructs we want to transfer into the plant via Agrobacterium. The second thing was, also in our Team V assay, we usually had 2 guide RNAs in a dual guide RNA approach, and more guide RNAs also increase the risk of off targets. And we were asking, do we always need 2 guide RNAs? What about if you only take 1? And again, to cut a long story short, it doesn't really matter in this case whether we have 1 or 2 guide RNAs, we see more or less comparable activities. And for a simple knock-in, which means you open a sequence and knock in something, we would recommend that 1 guide RNA is sufficient. But sometimes it depends what you want. If you want to replace a larger fragment with another one here in this moment, maybe it is beneficial to have 2 guide RNAs. We currently have some experiments to address these questions, but still in the process, we are still in the process of learning for this. Okay, another thing we wanted to know is, I mean, we have 2 exonuclease types, so T7 exonuclease and UL12, and I mean, in nature, there's 1,000 more homologs, and we wanted to dive into this a bit and screen for homologs of this. So you see a phylogenetic tree of a selection. So we have the T7 exonuclease group here. This is T5 exonuclease, and this is UL12 exonuclease group. So we compared the homologs of T7 exonuclease first, and we found a candidate which I called ME15. And here you see that it has more or less the same activity as UL12 in our assay. The benefit here is that ME15 is significantly smaller than UL12. And in terms, if you really are restricted in capacity of the construct sites, it might be beneficial. We also screened the UL12 type of exonucleases, the homologs, and here we found another exonuclease. It's from herpes simplex virus infecting papains, so that's why I called it PAP-E. And here we end up with double the efficiency, up to double, so in the palm-out orientation. So we have 2 homologs which are even better. So we have ME15 of the T7 exonuclease group and PAP-E from the UL12 exonuclease group. We, of course, wanted to apply our tools in real stable transformation events in which we changed the germline. And we had 2 approaches. The first approach was in Arabidopsis. And here we work with a special Arabidopsis line which carries a mutated gene, and we have to repair these mutated genes in order to let the plants grow. Without the repair, the plants don't grow and just die. So we can directly select for the desired outcome. And we did this comparing Cas9 with either ME15 or PAP-E fused exonuclease. And you see we got a few plants. So with 5 prime, also precise editing at the 5 prime junction, 3, 6, and 28. And of them, a few of them also carry the precise 3 prime junction is 1, 2, and 12. And if we normalize this to the number of seeds, we end up with a 10-fold increase comparing PubE fused Cas9 to Cas9. And the beauty of the system is that we can also identify transgene-free events, and among these plants, we found 2 plants which do not even carry the tDNA, so just performing the edit. Cooperation partners from Limagran also tested our tool in wheat, and here they did it a bit differently. So first they selected for transformants and then screened the transformants for edits to get an editing rate. And you see out of 87 transformants, we found 3 plants with editing events in the case of PubE fused to Cas9. And one of these plants were also precise on both sides. So if we calculate this, we end up with 1.2%. It sounds not so much, but it would mean that you have to screen 50 to 100 plants to get a desired edit. And we can see also the progeny, which is happily growing under bar treatment. So bar resistant was a desired outcome here in this case. So to summarize this a bit, we have an endonuclease, which is fused to PubE, and we use Cas9 intronized version, which was developed by Silvestre. And for the guide RNA, we use the flip extension scaffold, which was also published, and this is better for plant application. And I want this technique become a routine procedure, and I want to give you, use the last slides to give you some information of how you should design the donor, or we think how you should design the donor to maximize success rates. So here's just a gene, and imagine you want to have a desired edit at this point, and sometimes you not really find proper targets in close proximity to this edit. So what is a proper target? I also quickly summarize this here. So you know from the beginning, you have 20 nucleotide spacer followed by a PUM. And I tell you, for binding of Cas9 to a target, it's enough to have a perfect PUM and seed region, which are the first 10 nucleotides PUM proximal. So it can be all of the rest can be mutated, but if this is still the same as the on-target, Cas9 will bind there. For cleavage, of course, you need the complementarity of the whole 20 nucleotides. So when I say a good target is a target in which all of targets either have mismatches in the seed region or in the PUM region, the PUM motif. If you're not sure how the mutation of the PUM motif affects Cas9 DNA binding, I would recommend to use this PUM wheel here published by Chase Basil's group. So to cut a long story short, if possible, try to find on targets that have no Cas9 binding of targets somewhere else. What I also do then is to check splicing here. Therefore, we use NetGene2, so just put the mRNA sequence there and they give you some splice sites prediction. And here it's just about to not mess up the splicing architecture, which is there in the endogene, and not to put in artificial splice sites in our edit and changing the gene here. Yeah? So what I usually do is, once I found a good target, I try to replace the distance between the double-strand break and the desired edit with a synthetic coding sequence. The reason for this is I want to ensure the synthesis-dependent single-strand annealing, that after annealing of the 3' end to the donor, that this reaction goes really through to the edit. Because we know 250 base pairs identical sequence between the donor and the genomic DNA is already a super strong homology arm and can lead to abortion of this process. And then you have a conversion track that lacks the edit. So in order to force this stuff through till the edit, I recommend to put some mismatches here. And what I do is usually every 10 nucleotides, there will be a mismatch. Because we found out from the wheat data that sometimes a recombination window of 39 nucleotides can be sufficient to have this template jumping, for example. Yeah. What you also have to do here is, of course, to destroy the target, which you use to cleave the endogene, because you do not want to cleave and successfully integrate a donor. And here again, put mutations in the seed or mutate the PUM sequence. You could also check for splicing again. So as I said, we do not want to change these values here, but we also do not want to have artificial splice sites in our synthetic sequence. And if you have a donor site, you just have to mutate this GT here. And if you have an acceptor site, you just have to mutate AG to get rid of these unwanted splice sites, let's say. If you have this everything, then it's almost done. You just take the last and first mismatch and just take 250 base pairs in both directions as homology arms. Just a recommendation here, if your homology arm contains directed repeats, it's also a drawback because your 3' end can anneal in multiple occasions and you might end up with duplications. But if all is fine, you're ready, you have the donor, just take this donor, flank this donor with the target site you use to open the endogene here, and then you can synthesize this. Combine it with our other modules, for example, a state reaction marker, PubE-Cas9, or a guide RNA. You can do this with a MoClo system quite easily, and then you are done. Okay, to summarize this, I hope I could convince you that our exonuclease fusion to Cas9 leads to a significant increase in gene targeting efficiency, and with that, the UA12 type is always better than the T7 type. For the application, we recommend to use a single guide RNA and the homology arm lengths of 250 base pairs. PubE and ME15 are the best performing homologs. We tested, and of course, we have a patent application filed. And for the future perspective, we of course teamed up with many other scientists and want to get the system run in many other plant species, and for example, tomato, maize, and soybean, rice, potato, and more stuff is coming. So with this, I want to come to the end. So that's the team. So Alain and me, we developed this stuff together, supervised this project together. Sylvestre supported us with some nice reporter lines and all the MoClo stuff we use here intensively, of course. Anja and Petra, technician on this project, also Thomas even as a master's student back then, and the people from Lima Grand who do the wheat work. So with this, I'm at the end and I'm happy to take questions. Thank you very much.
Many thanks, Tom.